Hi, everyone. I'm Lauri Byerley, just so we get that on the recording. And I'm here with Dr. Ondrak and Dr. Gallivan, and we're going to talk about our project. Our title's a little bit different than the one that was advertised because about mid-year last year, we realized that we weren't really going to be able to do what we had originally planned on doing. And so we asked permission to switch, still doing the same kind of topic and everything, but a little bit different focus. So we're going to tell you about our project. That's the gut microbiome protein supplement study. That's what it's called with IRB and our research grant and everything. But you may have seen some ads. We've been titling it Protein and Gut Health Study. And we have developed this off of our previously published study. This came out in Nutrients early in 2022, and that study was also funded by APUS Research Grant funds. And I'm going to tell you a little bit about that study, which has led us to this current study. And then Kristen and Karyn are going to tell you more about the study, so we get to hear from all three of us. So the role of this current study is to identify a gut microbial signature of diets that are high in protein and purines in healthy young and middle-aged adults. From the study that we have published, what we did is we looked at people who self-reported that they were muscle builders, and they also either did or did not state, and this again was a self-report, that they took protein supplements. What we found in this group, the ones who were self-reported that they took protein supplements, is that they had a higher dietary protein intake. They had a higher fiber intake. This really surprised us, but that's the way it was. They had more fecal metabolites and bacteria associated with nitrogen breakdown, and they also had significantly higher levels of allotonin in their fecal matter, and this is a breakdown product of purines. While the ones who self-reported that they didn't use protein supplements, they had more genes associated with nucleotide metabolism and nucleotide synthesis in the fecal matter. These genes are coming from the microbes that are present in the feces. So the difference in diet and the gut microbiome and metabolome suggested that somehow protein, since protein is what we had a higher intake of, was influencing the gut microbiota. And we didn't measure the health consequences, but from the literature, we know that protein consumption leads to alteration in metabolites in the inflammatory response. So this allotonin that we saw, it comes from uric acid, and uric acid is an inflammatory agent. So we decided that we were going to explore this further in a second research study. And so the background for the current study is that, I didn't mean to move forward here, is that protein, the influence of protein on the gut microbiota is under study. We went to the literature and we did quite a bit of looking at the literature and what's out there, and there's really very little, and there's even less, and you'll see down here, purines on the diet as well. 50% of Americans consume more than twice the RDA. So RDA is their recommended dietary allowance for protein. So that, and realize that's dietary, it's not daily, but so more than twice what we really need in a day to rebuild our muscles and to maintain the protein stores in our body. And epidemiological data suggests that people who eat lower protein diets are healthier and have reduced rates of diseases like diabetes. And when I say the word lower, so they're closer to this RDA level. We're not talking about below, we're talking at least the RDA and maybe just slightly higher. Protein-rich diets also tend to have a lot of purines. And so that may be part of why we saw all the allotonin in the fecal matter of those self-reported muscle builders that were taking the protein supplements. We don't know. That's part of what we're looking at in this next study. And we also know there's volumes and volumes of information in the literature connecting the gut microbiota to many of the chronic diseases So that, in our mind, also warranted looking at this. So could it be protein that's helping to drive some of these chronic diseases? And that would be protein that gets into the colon where those microbes can have an impact. And so I want to just I can't neglect giving you a little bit of physiology, biochemistry behind all of this. So again, we have proteins and purines we're looking at. These are the two dietary constituents that contain nitrogen. That's what makes both of those unique. So we eat protein foods and purine containing foods. So these are going to be things like meat, fish, and don't forget we can get protein from plants as well. These are going to go into our digestive tract. In our stomach, we're going to actually break down some of those proteins into peptides and amino acids. Purines are going to pass through. These then go to this small intestine. The small intestine is going to absorb those amino acids, and it's also going to continue to break down the protein that wasn't and the peptides too. But the important thing here is we're going to absorb amino acids, and it's going to absorb some of those purines too. The amino acids can be incorporated into our skeletal system and a number of other things. The purines, there's 3 metabolic fates for that purine. It's going to be broken down to your uric acid. The uric acid could then go back, and this time it doesn't go to the small intestine, it actually is excreted into the large intestine. It can go to the kidney, or it can be converted in our bodies into allotonin, and it's going to be reactive oxygen species that do it. So the presence of allotonin in your blood can actually be used as an inflammatory marker. So in the colon, then you're going to get some of the protein that's not digested. So that ends up being about 17%, 20% of what you eat. Actually, it's just going to pass right through the small intestine, never be absorbed. And the colon can break that down into amino acids. And also those purines in the colon can be broken down to uric acid and allotonin. So that gives you a little bit of the physiology behind it, maybe more than you wanted, but I'm going to pass this on to Karyn, who's going to talk about the current study.
Okay, hi everybody. So our current research question is, how does bacteria in the gut interact with nitrogen-containing foods? So with that, a quick 3 bullet points of our methods. That's, our methods pages, our pages long actually, right? So we're analyzing microbes in our subject's poop. That's right. We are asking people to collect and send to Lauri our poop or their poop. And then after they take a protein supplement, that's 50 grams of protein. So any of you who use protein supplements in any way, shape, or form, you kind of have an understanding of what the consistency is, what it might taste like, especially if you only mix it in water. So we would analyze this after taking a protein supplement for just 7 days. Our participants they're randomized, and they're taking either a vegetable-based protein, pea protein in this case, or a dairy-based protein. This is whey protein. We ask participants to just continue with their normal daily lifestyle. So whatever they're currently eating, on average, please just continue with that. Even though these supplements, these shakes tend to make you kind of full, what we're trying to do is see what happens when there's an additional amount of protein in somebody's diet. So we try to make that pretty clear to them. All right, next slide, please. So now you start thinking about, okay, if I haven't done this already, and yes, there's a few of you out there, I believe, that have expressed interest. Now you start thinking, okay, how can I participate in this, right? So we're looking for healthy, active adults, right, over the age of 25. And then there's a bunch of exclusion criteria that you can read there. But a big one that we're finding is down in this bottom bullet point is if someone is taking antibiotics, because that does affect what's happening in the gut. So we wait at least 3 months. And we have at least 3 people on our list that we're waiting. In fact, the first person to do our survey, I think, was one of my nieces. She was just starting antibiotics. So in June, I didn't think we'd ever get here. I didn't think we'd need to wait, but we still need people. So in June, she will get to participate in the study, as will the others. Okay, so next slide. So once the total, the inclusion criteria is met and the interest level is there. Participants receive all of this, right? So they receive not just the pea or the whey protein supplement. They receive several flavors of each. We do have comments about these flavors. And they also receive the poop kit, as you can see a little bit there. So with pretty good instructions. And a nice shaker bottle. Here's the bottom of one. I don't know if you can see it here. Comes in handy. And then after the completion of the study and our surveys, they receive an Amazon gift card of $100. So obviously very nice. And then the study design itself, of course, once we see the boxes on the left, once we see the interested participants, we make sure that they're eligible. And once they are, and we start with them, they receive the kit that you just saw. And then Lauri, Kristen, or I were assigned as people's handlers. And so we contact them when they get their box of goodies and explain how they get started, and also send them links again to the surveys that they have to complete about their diet in the last 24 hours, and actually even questions about the poop that they're sending our way. So every day we reach out, remind them not to forget to take the supplement, ask them how it's going, answer any questions. And then as we approach the last day of the supplement, we remind them that it's time again to prepare for their poop collection. Once they do that, they repeat the 2 surveys and then sit back and wait for the results. So, all right. So I think next we will have Kristen continue.
Yes, thank you. So the recruiting so far, we've learned some things from the last study that we did. Here are our methods so far, and then we'll share what we learned. We have reached out through Faculty Connect, through social media, word of mouth. We've received a lot of participants so far from within APUS, which has been great. Emails sent to faculty and staff within the School of Health Sciences, the intranet, the Grad Hub. You can see emails to graduate students as well as the Research Fest. If for people that are interested in participating, they can click on this link. I also put that link in the chat when Karyn was showing you the inclusion and exclusion criteria. So we send that out, or they can also scan the QR code, and then that takes them to a Qualtrics survey where they can see if they meet our inclusion or exclusion criteria. And then if they're interested in participating, they're taken to a series of other questions. Next slide. So here's our progress as of the end of April. It's changed slightly, but this is what was our cut point for this presentation. We've had 94 participants attempt that survey to indicate or to determine whether or not they were qualified to participate. 83 completed all parts of the survey, and of those 83, 32 qualified. So again, reasons to be not qualified for the study: certain diseases, antibiotic use, certain, being too young. That was a common one as well. Our minimum age is 25. So that was kind of some common reasons for not qualifying. So of that 32, then 25 responded to the follow-up email Lauri would send to say, great, you qualify. Are you interested in participating? So we're happy with that 78% response rate there. 2 opted out. 1 got the kit but never started, so 8% there and 4% respectively. 2 didn't finish, and we blame FedEx for that. Lauri has been such an amazing, I'm sure she's learned more about FedEx than she maybe wanted to with getting some of these samples back, and the timing of the samples is quite key. And 2 of them were sent on time by the participants, but unfortunately not received in time to be processed. So as of, again, end of April, we have 15 completed. Next slide. Okay, so some of the lessons we learned and what we're doing a little different to try to improve participation, increase adherence, et cetera. As Karyn said, we have handlers. So once the participant agrees to participate and Lauri sends the kit, then we take turns reaching out to them as their contact person, asking if they have any questions, reminding them what to do on each day, asking if they, if they need a link to one of the surveys, the dietary survey or the physical activity survey. So I think it's that daily communication that has really helped. Some participants prefer text, some prefer email. We kind of go with however they are responsive. We've also compiled a list of recipe ideas for both the pea and the whey proteins, just to give them some variety. And we try to reassure them they can take any flavor. It doesn't have to be one flavor. And the company was kind enough to, again, supply 3 different types: unflavored, vanilla, or chocolate. If they report any sort of issues, we try to walk through, all right, maybe you can try to break it into 2 doses or other problem solving, troubleshooting like that. Next slide. And so here are some of the participants' comments we've compiled. It's very easy to take the protein. I appreciate the check-in. Awesome, thanks. It's going well, no issues. Chocolate is the go-to. I'll let you read the rest of them, but we did find we have some interesting feedback. Some people really feel strongly about one flavor over another, but I would not say that's unanimous. There is not one favorite flavor. So again, we feel fortunate we have more than one. So yes, so far, lots of positive feedback from our participants. And again, in the chat, you will find that tiny URL if you or someone you know might be interested in participating, or at least seeing if you qualify.
Also, before Samir has a question here. You are welcome to share that with anybody. Anybody who qualifies can take the study. So why don't you take that with you tonight and pass it on to your friends. So the question Samir asks is, how does high protein intake, the supplement, affect fermentation in our body? How about those who have IBD and other gut issues? Can they participate?
So the answer is, at this point in time, we're trying to study, as I said, healthy individuals, or Karyn said. So if you have IBD, for example, then you'd be excluded, because again, we need to make sure that we have a little bit more homogeneous group. So yeah, that you wouldn't be counted in. Fermentation is a process that happens in all of our guts. And so yes, that protein is being fermented in the colon and the purines too. And I should say that the whey supplement has no purines in it. The pea protein does have purines in it. Let's see, Stacy asked, determine which communities, how do you determine which community? So you're
that was for
okay.
presentation.
Yeah, the question, Lauri, is what is your goal population number?
So we used, we're working with a statistician, and we did do a power analysis and it was based on the allotonin difference that we found in this previous study. And from that, we came up with needing 15 people, at least 15 per group. So we are gunning to get 15 per group. We are a little more than halfway there. Some of it will depend too, because everybody is randomized. You have no choice which one you're going to be in if you decide you want to participate. That's all been predetermined by a randomization. And that randomization isn't perfect, so we probably will end up with a few more subjects than the 15 per group, but we need 15.
Look at that, the last one of the day, and we finished early.
Well, you're welcome to ask questions. Come on, I can ask questions too.
This is your time for questions. For
Yeah.
sure. So you all are continuing this study then, right? Still ongoing. You're still accepting participants for exactly what you said, right, Lauri? Like another iteration or another like measuring something else, if I'm not mistaken.
So we will continue to recruit subjects until we get our number. We can go until December. We're hoping that we'll have everybody by the end of the summer that we need. All the samples are currently in storage, And then we will send those out for analysis. And so the analysis that we will be doing on the fecal samples is the gut microbiome, which will determine what gut microbes are present, particularly bacteria. And then the metabolites, we will also look at the metabolites. There's a couple other analysis we may do on those samples, but we'll wait and see where we're at. This data, I should tell you, generates a lot of in what we're dealing with, they call it artifacts. So the gut microbiome, we will see anywhere between 600 and 800 species of microbes. For the metabolites, we will see anywhere from 200 to 1,000. It just depends on which analysis we're able to do. A little bit comes involved in that is expense. For the dietary information that we're obtaining, we have about 300 to 400 data points there. It's very detailed. And then we have the International Physical Activity Questionnaire. So it's a pretty massive database that's built off of this, the collection of these samples from all of these people. And so that in itself will also be a project, is getting it all analyzed, but we are working with a statistician to make sure that we do that all right. We are not looking at gender or ethnic differences because we do not have a large enough sample size. That would require thousands of samples to look at, and we don't have the bandwidth to do that. What was I missed? There was one other question here. Would you like to review, is that one for us?
Yes.
Exactly what you're looking for again?
Yes, which I think you.
Oh,
But yeah, I believe you already answered it. Mhm.
Yep, already answered.
Oh, what was the number of kits results received compared to the kits that were requested
Oh.
and shipped? So we have worked very, very hard. We learned from that first study. The first time we sent out this announcement, we had a lot of people that filled out the survey. And so we went through and we figured out, okay, who qualified and who didn't qualify. And there were about 60 people that qualified. So we immediately shipped them all kits. Well, then we didn't get 60 samples back. And like, well, what are they doing with our kits? And, you try to track them down and find them, and we couldn't, and they wouldn't respond. So that's why, Kristen talked about what we do now. So we send them an email that says, okay, you did qualify. Are you really interested? And if they respond back to us, then it's like, okay, we have somebody that's responsive. So now we'll send you a kit. So let's see, we had one individual that responded to us and we sent them the kit and we never heard back from them. But most of the people, once we do that and we establish some kind of communication with them, they're responding back to us. And we're sending them the kit and we get the samples back, they complete the study. Unfortunately, FedEx, if you remember back in, let's see, was it March, Karyn and Kristen?
Oh, we had weather issues and different things.
Yeah.
Yeah.
And those weather issues screwed up a couple poop samples, and they have to be received back in 24 hours overnight.
Daniela, they also have a kit project. So yeah, we've only, as Lauri was saying, we only had one person who got a kit and didn't start the study. So not a lot of loss from that perspective.
Yeah, which actually we're really excited about that. We're amazed. So we are a little top heavy. We got lots of kits. So fortunately, I don't think we'll run out of kits. Yeah, Daniel, be sure and think about how you're going to optimize or minimize, I should say, the loss of kits. So how many, in 2022, we were really getting organized for the project. So we got IRB approval. We started with it a little bit behind the ball, but we got it going. And then 2023 was when we were going to always recruit. And we started recruiting the end of February, the beginning of March. Yeah, I'm trying to think when we sent that first one out. And so we've been recruiting since then. So it gives us the whole year to recruit. We do have the whole year that we could recruit, but we're hoping we'll get it done before then. Does that answer your question? And always, Danielle, always anticipate it's going to take you at least twice as long as you think. We learned that too from that first study. We ended up having to do 2 cohorts for the one that we published. And we really needed to go for a few more people, but then we ran into COVID. And so we had to shut it down with COVID. And actually, I'm glad because, of course, we know that one of the ways that they monitor for COVID is to go to the sanitation departments and stuff. So it's in fecal matter.
One thing we learned as well was all those recruitment methods we shared, Faculty Connect or listservs. We didn't do them all at once. We tried to space them out and, maybe hit this population first, deal with the influx of potential participants, and then go on to the next one. So we've tried to do that systematically. So we still have some populations who we haven't reached out to yet to try to get that second half.
Yeah, and Karen's really been our marketing guru, and we do have a plan. So we're going to be down right now. We're still recruiting, but we won't start collecting samples until towards the end of May again. And we've got a whole game plan for June and July to try and really push and get the last number of participants we need.
With that, I don't see any more questions coming in. For some reason, mine are being filtered by wherever somebody's time zone is right now. So that's fun. I want to thank you all so much for your time. This is really excellent. I love hearing the progress on everything. I'm sure we'll have another presentation next year to touch base and see how things are going. So thank you all also so much for joining us today, popping in and out, seeing what everybody has going on. It's a real pleasure to be able to host our researchers as they report on what they've got going on and to see everybody. So thank you all again so very much.